Let's now consider the semi-permeable capabilities of the plasma membrane and why it is designed specifically to be so. Now, as I've mentioned earlier, smaller molecules such as carbon dioxide and oxygen, even water, they have the ability to pass through the plasma membrane freely, right, without requiring any additional help. And this would be even better if these molecules are hydrophobic. That's because they need to pass through this hydrophobic layer. So hydrophobic molecules, small hydrophobic molecules, usually do not have any issues passing through the plasma membrane. They do not require any additional help. But larger substances like sugars and amino acids and other larger molecules, they can't just simply pass through. So the membrane allows certain things in, but prevents everything else. For that reason, the plasma membrane is said to be semi-permeable. Now, some cells, they may have channels for water to pass through. Now, even though water has the ability to pass through the plasma membrane on its own, some cells, just to speed up the process of water entering and leaving the cell, they may have specific protein channels called aquaporins. Now, aquaporins were first discovered in human red blood cells, right? RBCs. They were first discovered in red blood cells, and then now we know that certain types of bacteria also have it, and E. coli was the first bacterium to have that was discovered to contain aquaporin. So the aquaporins are channels that allow water molecules in and out, and it's believed that they are present basically to speed up the process. In terms of molecular movement through the plasma membrane, we also have simple diffusion. For example, when we said that oxygen and carbon dioxide, they can go in and out of the plasma membrane freely, that is largely due to diffusion, right? Which is defined as the movement of molecules from a region of high concentration to a region of low concentration. So oxygen and carbon dioxide, they would move through the plasma membrane depending on their respective concentration gradients. So wherever oxygen is higher, it moves away from that to somewhere where the concentration is lower. So that is your simple diffusion. And of course, osmosis is specifically the movement of water molecules, again, from a region of higher water potential to a region of low water potential, right? Now, the movement of water molecules is, as you know, it's dependent on how much water is present inside or outside the cell. So we use three specific terms to describe the concentration or the availability of water across the membrane. Now, hypotonic solution. Now, this is a cell, all right, this is the cell, and this is outside the cell, okay? Now, if the cell is placed in a hypotonic solution, now, as you know, solution by definition is a mixture of solute plus solvent. All right, you take a solute and you dissolve it in a solvent, you get a solution. If you dissolve some sugar in water, then you have a sugar solution. If you dissolve some salt in water, then you have a salt solution. So a solution is basically a mixture of a dissolved substance and a solvent. Now, if you place a cell in a hypotonic solution, it means that the amount of dissolved substances outside the cell is lesser than the amount of dissolved substances inside the cell. So here it's greater. All right. Now we have to look at the purple circles. The purple circles are the dissolved substances. So if you look at the hypertonic solution box, you have fewer dissolved substances outside than inside, which means the amount of water, all right? So here you have more water than here. All right, because if you have lesser dissolved substances, that means your solution is largely solvent. So you have more of solvent than solute. And here solvent is referring to water. So you have more water outside than inside. So water would go from outside to into the cell, right? Water flows into the cell. So when water flows into the cell gets larger and larger, it starts swelling up. All right, the cell starts swelling when it gets larger and larger. Now, in the case of bacterial cells, as I mentioned earlier, they have cell wall, right, which our cells don't have. So as the cell gets larger and larger, the cell wall protects the cell from bursting. This is the same thing that happens in plant cells as well. The plant cell wall prevents the cells from bursting. But in eukaryotic cells, because we do not have these cell wall, so if our cells consume too much water, which means if too much water enters our cells, then our cells will end up bursting, which is a simple experiment that we can do with red blood cells. We can place some red blood cells in a hypertonic solution, and when you look under the microscope, the cells would have burst because too much water enters the cell and there is nothing keeping the cell intact. Now, the second situation is if you were to put the cell in a hypertonic solution, situation 2. So if you count the number of circles outside, you have greater number of dissolved substances outside than inside. So what about the amount of water? If you have a lot of dissolved substances outside, that means the amount of water is lesser outside than the amount of water inside. Okay, so less dissolved substances means more water. More dissolved substances means lesser water. So because the amount of water inside the cell is greater, so I've got 2 ticks here, than outside, where I only have 1 tick. So the amount of water, the direction of the water flow would be from inside to outside. So water starts leaving the cell. Okay, water starts leaving the cell. And when water starts leaving the cell gets smaller and it starts shrinking. Now, when it starts shrinking, the cytoplasmic membrane, right, the plasma membrane, would start detaching. It will start pulling away from the cell wall, right? It pulls away from the cell wall, and this condition is called plasmolysis, right? Plasmolysis is a condition where the cell starts shrinking and the cell membrane starts pulling away, detaching from the cell wall. Now, how about isotonic solution? Isotonic means the amount of water outside and inside is the same. Okay, so there is no net flow of water in either directions. All right, it means the amount of dissolved substances are equal on both sides, which makes the water potential equal on both sides. Therefore, there is no net movement of water molecules in either direction. So that's an isotonic solution. So hypotonic means the water potential is greater outside, so water flows in. That is box number 1. In hypertonic solution, it means water potential is greater inside the cell than outside, so water flows out of the cell. Box number 2. In a hypotonic condition, in box 1, when water enters the cell starts swelling, and it's prevented from bursting because of the cell wall, the powerful cell wall. In situation number two, when too much water leaves the cell, the plasma membrane pulls away from the cell wall, right? And a condition that is called plasmolysis. Now, the electron transport chain is a very unique function of the bacterial cell wall. Now, I'm going to digress and go a little bit into biochemistry. Now, in eukaryotic cells, in our cells, um, the organelle that produces energy, ATP, is the mitochondria, right? So ATP is produced in the mitochondria, specifically in the membranes of the mitochondria. Mitochondrial or membrane, the membrane of the organelle contains protein complexes that will produce energy, ATP. Right? I'm not going to go into too much specific details there. But in the case of bacterial cells, in prokaryotic cells, they do not have these organelles. Prokaryotic cells do not have membrane-bound organelles, which means they also do not have mitochondria. How else will they then produce ATP? Now, in bacteria, in prokaryotes, the proteins that produce ATP in the mitochondria, they are present in the bacterial's plasma membrane. So in eukaryotes, these protein complexes are present in the membrane of our mitochondria. But in bacteria, because they do not have mitochondria, these protein complexes are present in their cell membrane. All right, their functions are pretty much very similar. Right, these protein complexes, they function pretty much in a similar manner, and that's how they produce ATP. Now, one such a process which bacterial cells have to produce ATP is this thing called the proton motive force. Now, what this system does is it uses energy from electrons to pump hydrogen ions out of the cell. Okay, that's why it's called the proton motive force. So the hydrogen ions are pumped out of the cell using energy from electrons, because to pump it out requires energy. Now, once hydrogen ions have been pumped out of the cell, then what happens to the concentration gradient? You have more hydrogen ions outside the cell than inside. So hydrogen ions would start flowing back into the cell. Let me see if I've got a better picture. No, I don't. So they would start flowing back into the cell. Now, once they start coming back into the cell, I'm going to use this very simplified drawing. Now, so that is the outside, and this is the inside. Now, the hydrogen ions will start coming back in. The H plus ions will start coming back into the cell via a specific protein channel called ATP synthase. ASE. So the hydrogen ions will start flowing back into the cell via this protein channel called ATP synthase. Now, when hydrogen ions flow through this protein channel, it will result in the production of ATP. So you have a lot of ATP being produced, and that's how these cells they produce energy. So they first pump the hydrogen ions out, so the hydrogen ions would accumulate outside the cell, and then by concentration gradient, or they call it an electrochemical gradient, the hydrogen ions will start flowing back into the cell, and they flow in through a specific protein channel called ATP synthase. So the flow of the hydrogen ions from a region of high concentration to a low concentration results in the production of ATP. So this is quite an interesting mechanism that they have, the proton motive force. So they use the movement of the protons to actually produce energy. Now, movement of small molecules, especially these hydrophobic molecules like nitrogen gas and oxygen gas, they move very freely, right? Now, through the plasma membrane. But sometimes other molecules like amino acids, they may require special transport proteins, right? They're called, they can also be called as carrier proteins or permeases because they permeate through the plasma membrane. They allow these molecules to go into the plasma membrane, and they are what we call membrane spanning, which means they are present across the membrane, right? The entire protein molecule is present across the plasma cell membrane. So one region of the channel is facing the outside, and the other region is facing the inside, right? It's just like how you have these underground passages, right? So it takes you from one region to the other. So one end of this underground passage is exposed to the outside. Likewise, the other end is exposed to somewhere else. So it takes you from one place to another. So um, that's what these transport proteins do. And but these transport proteins are very, very specific, right? Which means they are only designed to allow a specific type of molecule in or out of the cell, right? So you have different types of carrier proteins for different types of molecules. So if you look at this cartoon, this channel, this protein channel, purple channel, is apparently only specific for these orange circles, these peach colored circles. The reason is because once the peach colored circle, once this circle enters the protein channel, right, it binds to an active site in the protein channel that will allow the protein channel to change shape, right? So once it binds to this particular region, it's like a signal, and the protein channel would change shape. So as you can see, it went from a V shape, right? So this is like a V shape where the opening is where the top of the protein channel is open to the outside but closed to the inside, right? Goes to an From there it becomes an inverted V, where now the outside is closed, right? Outside is closed, and the inside is open. So the orange circle can enter the cell. So this is a very clever way of making sure that only certain molecules enter the cell, because these protein channels are very specific to which type of molecules they allow into the cell. Because the binding of the molecule to the protein channel, the molecule first has to bind to the protein channel, and that will cause a conformational change. The protein channel undergoes a change in shape that will then allow the small molecule to enter the cell. Now, the different types of movements that we will encounter in bacterial cells. Now, a lot of these are also common in eukaryotic cells. In bacteria, we have the process of facilitated diffusion, which is basically a type of diffusion where molecules move from a region of high concentration to a low concentration, but sometimes they require the help of additional protein molecules. Okay. Now, I'm just going to use this diagram to basically explain everything. So the first one is facilitated diffusion. Okay, so if you look at these orange circles, you have a high concentration of them outside, then inside. So naturally, they would flow down the concentration gradient, right? So this process does not require energy. Okay, this one doesn't require energy, no energy required. because this is a passive process. It's happening due to its own concentration gradient. So the orange circles are moving into the cell down their concentration gradient. The green channel is simply there to facilitate the process, to make it happen quicker or better. Apart from that, this is a non-energy requiring process. Now, active transport and group translocation, these are processes that do require energy. Okay, they do require energy. So, for example, in this case, we are looking at a proton motive force. So, hydrogen ions, right? They're coming back into the cell. So remember, I told you in proton motive force, first the hydrogen ions are pumped out of the cell so that the concentration increases outside. And then when these hydrogen ions start flowing back into the cell, they produce energy. And this energy is then used to bring in other molecules. So if you look at the green boxes, there are fewer green boxes on the outside than inside, right? On the outside, if you look at this cartoon, there are only 3 green boxes. But if you look inside the cell, you have 1, 2, 3, 4, 5, 6, 7. So if you want to bring the green boxes into the cell, this is going to be difficult because the concentration is actually lower outside than inside. So bringing them from outside to in, it means you're going against a concentration gradient, right? Unlike in diffusion, where it's a passive process, it goes down the concentration gradient. But now you're going against the concentration gradient, which means it's like you're swimming against the current, right? If you're swimming along the current, then it requires very little energy input from you. You just have to float and the water takes you down. But if you're going to swim against a current in the water, then you have to fight against the power of the water flow. So that's what's happening here. You're trying to bring molecules against a concentration gradient from low concentration to high concentration. So that requires energy, right? So here in this case, the energy is provided by the proton motive force. The influx of hydrogen ions into the cell will produce energy, and that energy would be used to bring the green boxes in. So the bringing of the green boxes in actually requires energy because this is going against the concentration gradient.